So, just to be very quick, this is just a few slides for those who are not familiar with the whole exome and whole genome sequencing, and so just I apologize for those who are familiar. Okay, so just to remind you something. In fact, in the genome there are 30,000 genes, and the whole exome corresponds to, in fact, the part of the genome which is expressed and they're translated. It's 50 million base pair. And when you are talking about the whole genome, it's 3,010 to the 6 base pair. So the expressed sequences represent only 2%, something around 2% of the whole genome. If you do a whole exome sequencing in any one of you will find 35,000 variants if you compare to the reference human genome, which is in the database. So if you do the whole genome sequencing, you will find 4 million variants in each individual. So just to position the problem. How do we identify genes with whole exome sequencing? In fact, basically, it's a stepwise, I guess, this morning, probabilistic approach. And this approach is not black and white. In fact, basically, I will just squeeze completely the technical parts, leading to the enrichment, capture of the exons, etc., but which are very important. And after that, I will go to how we filter. First, we have to work on high confidence variants. Many variants are not high confident. That's the main problem. And after that, we have to, depending of the pattern of inheritance, and sometimes you are not sure of the pattern of inheritance, so that's a problem in small families. You will filter the variants that you have identified, and also you will have also the frequency of these alleles in general population in databases. Oh, mais c'est pas vrai. In databases, and but sometimes these frequencies are not the ones that you have in your population, for example. To make a long story short, in fact, once you have done your whole exome sequencing on a good platform, in fact, what happens is that you have to filter, what is shared by the affected members of your families of interest. And after that, you go to databases and you try to find rare variants and to select rare variants. So, as you say, it's probabilistic. And just to give you an example, here, I've taken a slide from Edgar Verdura in the lab, and this is one of the families that have been shown this morning, and the gene is not yet identified. And you see that it's this family here, and you have two here brothers, and I should say here you have one brother, one sister, and here you have another brother from the same mother. The mother was affected, and all three. So we have two half brothers whole exome sequencing here and here. What can we expect? Basically, each time you do a whole exome sequencing and you filter for rare variants below 1 in 1,000, you end up around 350 variants in each individual. So you have to realize that that's a lot. That's a lot, basically. So you have to filter, and you can imagine immediately that as the patient has to share the affected variants with the brothers, that divide enormously. So if you have an additional member, it divides again. So that's very important. Let's show this example. Here, a family, toujours by Edgar, and you have 3 individuals who have been collected and had the whole exome sequencing. 2 brothers. I'm sure Lucie will recognize this family in the room, Lucie Guillaume-Maréchal, and this is a cousin. And you see, instead of 56 variants, we end up with 5 variants. So that makes a huge difference. That's why we worked with Dominique and Nasira. Okay, so that's the way to do it. And so it is a powerful approach somehow, but it has limits. In 2012, there have been 100 genes causing Mendelian disorders that have been identified with whole exome sequencing. But the majority of these genes are genes involved in autosomal recessive disorders, because the method is much more powerful for recessive disorders than for dominant disorder. But still, there were around 40, a little bit less, genes involved in autosomal dominant and X-linked disorders. The problem is that there was a review recently saying that at least 50% of whole exome projects fail. And they fail because of very known reason now: incomplete coverage. Many sequences are not covered. The small insertion deletion are not detected. Large copy are not detected. Obviously, mutation in intronic not expressed sequence are not detected. And so it's not easy. So what about whole genome sequencing? Whole genome sequencing, it's interesting because the coverage is much better for technical reasons. The mutation in intronic are detected. The copy number is feasible based on the data of the sequencing. Therefore, it's a good approach. The problem is that, you see, there are very few genes that have been identified with whole genome sequencing. And why is that? It's a bioinformatic challenge. You have to deal with 4 million variants for each individual. So imagine the huge amount of data, the analysis, the storage, and these very few academic labs for the moment do whole genome. So this is one of the challenges for the next 5 years. Alors, going back to a small vessel disease, we really need to go further and to analyze very large number of independent small vessel disease if we want to analyze or to identify the gene involved in sporadic cases caused by de novo mutation, sporadic cases which are genetic but they are recessive disease. In the stroke field, almost nobody has worked on recessive disorders. The families are small, and so generally there's only one case per family, so the clinician will think that it's not genetic, so they don't just collect the patients. Need for this whole genome if whole exome is negative, because we are going to discover now the mutations which are not in expressed sequences, the deep intron and reorganization of the genome, etc. So this will be done with whole genome sequencing as well as a technical approach. But we have to solve this problem. We have also to solve this problem, and this problem will help for this problem, is that we really need, for example, to give you an example in the lab, we have some patients now in the diagnostic lab which are sent and required call for A1 genotyping, call for A2, Notch3, TREX, Carazil gene, HDR1, we have not yet set it up. And I mean, no way, I mean, if we have to have technicians working days and night by the Sanger sequencing, we are not going to be. And on top of it, you look only to, you find only what you look for. If you do an exome sequencing, you can also look for other genes. So our goal in the hospital here now is that. And that's not an easy challenge, but we have to do it. And obviously, the next challenge would be to identify the mechanism, and this is multidisciplinary approach. Going back to the geneticist, in fact, there's another challenge which I think will be important and will be tractable. It's not tractable for the moment, but it will be tractable with this whole genome sequencing, whole exome sequencing, is the identification of the protective variants. I think it's extremely important. In dominant disorders, many of them are incomplete penetrance disorder. And in fact, it would be very interesting to understand why in a family you have guys who have exactly the same mutation, and one would not be sick and the other one would be sick. Or one would not be sick until, let's say, 80 years old, like I saw with Hugues in 2 families, and in fact, the other one would be sick at 45. And this is extremely important. This for therapeutics after that would be important. And I think that I think it should be good.